Here are the primary players involved in bacterial DNA replication. Okay. Rule of thumb. Okay. Side note here. Rule of thumb. When you're looking at a textbook, you're looking at a figure from a textbook, or you're looking at a figure from a paper, and you're not sure if they're talking bacterial or maybe eukaryotic mammalian DNA replication. Look at how they designate the polymerases. You see here, it's Pol 1, Pol 3. Okay? There's like 7 different polymerases in bacteria, and they are numbered 1 through 3. Each has a different job, different role, does different things. Since they are numbered, Roman numerals 1, 2, 3, 4, 5, 6, 7, it is prokaryote. If it's Greek letters, alpha, beta, DNA, polymerase beta, alpha, things like that, that's eukaryotic. Okay, the eukaryotic has many, many more components, is much more advanced, has better proofreading abilities and things like that. So we generally in textbooks just discuss the bacterial because it is simpler and it's a little more well studied because we can go through and we can mutate the specific gene in a DNA chromosome, whereas a eukaryote, we have to worry about the fact that all genes, are going to have 2 versions, 2 alleles, because there's 2 sets of chromosomes, and there could be multiple copies of the same gene spread out on different chromosomes and things like that. So we do a lot of our studying in bacterial models, and then we just find homologous enzymes, homologous processes in eukaryotic cells. So when you look at it, the big picture, replication fork, leading strand, lagging strand. As I said, leading strand, lay down 1 primer, DNA polymerase just runs. As helicase unwinds, DNA polymerase is right behind it. Go, go, go. Lagging strand's a bit different. You're going to have you're going to have the primase, the RNA primer, laying down RNA primers, running along behind the replication fork. Every few dozen, few hundred nucleotides, new primer, new primer, new primer. DNA Pol 1, or I'm sorry, DNA Pol 3, doing that reverse leapfrog thing. Remember the reverse leapfrog, lay down, new here, and then jump back to this one, lay down, jump back, and so on and so forth. DNA Pol 1 comes in, removes the RNA primers, replaces it with DNA nucleotides, and then DNA ligase just comes along and glues everything together. When we draw it out, and we make videos, little cartoon videos of this, we have always drawn it so that the enzymes run down the double helix. Turns out we're probably wrong. New research has shown that the enzymes and the proteins that are at work in the replication fork are actually most likely stationary. And it's the DNA that's pulled through the stationary enzymes. Lamina propria, remember, is the cytoskeletal structure inside the nucleus, maintains the shape of the nucleus. Also gives the enzymes a place to anchor to. So that way the enzymes are in one place, and it's like you're pulling a string through the enzymes. The enzymes, the proteins form this one giant complex, this globular thing, and then you're pulling the DNA through. As the DNA is pulled through, it's copied. It's pulled apart, and two new strands are made.